So biology is like 1665. Robert But Hooke. He discovered that cell look like honeycomb structure, some shit like that. This is the guy who cooked this thing. Honeycomb structure. Then the Leeuwenhoek, the Leeuwenhoek, he did was he took a pond water and looked at the microorganism inside of that pond water, like bacteria and living cell. He discovered bacteria and living cell. Then you have Robert Hooke, Leeuwenhoek. Leeuwenhoek was 1674. Then you have cell theory by Shandon and Schwann, double S brother. All living cells are made up of cell, and cell is bend like the fundamental unit of life. Cell theory. Cell theory was given in 1838. Theory like all living organisms are all living organisms are like made up of cell and all that. This theory is given by double S brother Sheldon and your Sachuan. These are the two guys who gave this theory, Sheldon and Sachuan. Then there is your Rudolf Virchow. All new cell will arrive from pre-existing cell. It is given there. Uminous cellular is cellular. So first of all, it is Robert Hooke, the comb guy. Then there is Leeuwenhoek, a 1674. He's the guy who told you about like your. He's the guy who told you. Like he took a pond water and looked at the microscope. Then there is two S brother in 1838, the Sheldon and Swan, who said that all organisms are made up of cells and all that. Then there is your Rudolf, like a wolf, Rudolf, Wischkow, Rudolf Wischkow in 1855. He told that this cell is made up of pre-existing cells. Yeah. Then there is your prokaryotic and eukaryotic. Prokaryotic have these are prokaryotic seems pro, but they are not pro. They are no. No nucleus, cell membrane is there. Cytoplasm, genetic material of no nucleus is there. DNA deoxyribonucleic acid. Then cell wall, some prokaryotic, then unicellular, then single chromosome, mostly small size, no membrane. Then eukaryotic have nucleus paired chromosome, so they have 23 paired chromosome. Like some have like ribosome, ATS, and some shit like that. And the smallest cell is overall mycoplasm gallicitium. Large cell multicellular unicellular to like amoeba produced by endoplasmic reticulum for Golgi bodies. Separation of membrane or formation of Golgi bodies, they transport into different. Then there is a cell wall, sorry, cell membrane. Cell membrane is the outer layer of membrane, extra rigid, hard, dead skin, semi-permeable plant. Okay, now you have your cell membrane. Cell membrane is like the outer membrane of a cell, this kind of thing. So this kind of cell membrane is like extra rigid, usually dead skin to protect from the leaking of water and like from other like harmful in substance from the environment and all that. Then it is semi-permeable, like it is dead skin. So dead skin is not really that attached to each other in comparison to a living skin. So it would have some pores in it, so it just leak water from there, but the inside one is the one which holds the water. But plants do need to lose water over time. Then in cell membrane in different plants is like it in plants it is called your cellulose, but in fungi is it's called chitin, and in bacteria is called peptidoglycan. Peptidoglycan. So plant it is cellulose, fungi it is your fungi chitin, then bacteria is peptidoglycan. Plant cellulose, fungi chitin, then bacteria peptidoglycan. Then plasma membrane. It is defined as cell region, living, flexible, selectively permeable, lipids, protein, fats, pores, size factor, concentrated higher to lower osmosis, difference of water in liquid, then lower to higher reverse osmosis. So yeah, this was cell membrane, the outside one, but the inside one, which is the main protective one, which holds the internal organs together, it is your plasma membrane. And plasma membrane is it is defined as cell region, living, flexible, and selectively permeable. This thing is living, not dead like your cell membrane, whereas cell membrane, the plants have cellulose, the bacteria have chitin. Chitin, the no bacteria, not bacteria. Fungi have chitin, bacteria have peptidoglycan. But plasma membrane is living and it is flexible. If it is living, then it is flexible, not rigid like your cell membrane. Plasma membrane is flexible. Then, if it is flexible, the plant, like your uh, cell membrane, do need to lose water. So when it loses the water, it should be relatively selectively permeable, preamble, whatever it called. I just call permeable. Uh, Preamble. Okay. And lipids, fat, protein is used to make this, I guess. Lipid, fat, and protein. I would say it is basics to like have fat or protein in a skin, I would say. Then pores are also there. Connective predicament. Higher to lower is osmosis. Lower to higher is reverse osmosis. So pores. So like the removal of water is a very common thing in the plants. So plasma membrane helps in that. So like the pores, which are the holes in this plasma membrane, or like you could say like this is the skin and this is the hole, it could get expand this much. So the pores, when it get expands and release the water, is called osmosis. When the pores are small and it won't remove the water, it don't even want to remove that much water, like your cactus skin, it does reverse osmosis. Then there is your protoplasm, pre-Jenkitum, physical basis of living William Huxley. Then now plasma membrane. Medium solution is equals to solute plus solvent. Medium of plasma membrane, hypotonic solution, isotonic solution, hypertonic solution, RNA, protein synthesis. Does plasma membrane have DNA? So like this plasma membrane can like shrink and expand on basis of solution inside present of like whatever the stuff or the thing inside of it is. Like if it is like hypertonic solution, what it will do, hypertonic solution, is that the solute is more than your solvent. Like solute is more than solvent is the water, solute is like supposedly this is a bottle of ruabza, right? And in this ruabza, till here the ruabza is filled with the high concentrated ruabza, and like you are filling it with this much of water, so your solute is more, sorry, so the solute, the ruabza is more than your solvent, which was the earlier water. In this case, a cell wall will undergo plasmosis if it is placed in which solution. So the cell wall in this case would go under plasmosis. So like supposedly this is the cell and this is a container. Now if you have more solute here and less solvent, then the like more solute is there, right? The more concentrated stuff is there, right? Imagine you have more concentrated stuff and you have a less concentrated body inside of it, so it would suck out of its water and all that for here. So it would just like your wait. In hypertonic solution, does the cell inside the container swells up or swells down? So, like in a hypertonic solution, where the solute is more than solvent, which is the water, so there is more concentrated stuff than the water inside a container where is a cell inside of it. So, like the cell would swell up or swell down.
In a hypertonic solution, the cell will typically shrink or shrivel. This is because there is a higher concentration of solutes outside the cell compared to inside, causing water to move out of the cell through osmosis. ये सिर्फ एक सामान्य जानकारी है
Okay,
और ये
so like it is going outside and the cell would shrink, right?
Yes, that's correct. Water will move out of the cell, causing it to shrink.
And osmosis mean the ability to remove the water from inside of it, right?
Osmosis is the movement of water across a semi permeable membrane from an area of lower solute concentration to an area of higher solute concentration, which often results in water moving out of a cell in a hypertonic solution.
Yeah, so I was just got confused with the diagram. My diagram is showing like something like this in the my notes. So this arrow outside is water, and when the water is coming outside of the cell, and this is doing osmosis because water is coming outside. The difference of water liquid, that is the whole thing. Osmosis is happening here, and the whole thing would shrink down like this because the water have been sucked out of it. Then there is your in this thing the plasmosis also happen. A cell will undergo plasmosis under which of the solution it is your hypertonic solution. Then there is your isotonic solution. Isotonic solution is the is like you have same level of solute and solvent, the whole thing. The solid is water. Ugh. And the solute is like some supposedly like ruabza. So like this is the concentration is same, so there is no force applied on it. This is isotonic. Then there is hypertonic and hypotonic. Hypotonic. Yeah, here you are drinking hypotonic. So here you are drinking hypotonic. That's a brilliant thing to say. Like this is here. Here the water is going inside because the cell have more solute, and it is just sucking up water to make it like normal, so it would swell up. Hypotonic, you are drinking a tonic. Hypertonic is like you are rejecting the tonic, it became too powerful. What is R? What is RNA? Ribonucleic acid, a single standing nucleic acid essential for life and bridge between DNA and protein. Does RNA mean DNA? Does RNA mean DNA? Ah, so they are the single strand one. Okay. RNA is equals to protein synthesis. I know that much. Rough endoplasmic reticulum. Ouch, 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 ouch. So these all have your uh, RNA, which is your little brother of DNA, which is like the one with, what can I say? The RNA is like DNA is this DNA, this double strand one which have like this kind of structure. This is DNA. RNA is like this. So nucleic, so nuclei, cytoplasm, mitochondria, chromoplast, and ribosome have RNA. So I'm studying here that nuclei, cytoplasm, mitochondria, chromoplast, and ribosome have RNA. So don't they have DNA?
You're right that all those things have RNA, but they also have DNA in different places. The nucleus is the main storage place for DNA. Mitochondria and chromoplasts also have their own smaller amounts of DNA. Cytoplasm contains various DNA molecules, and ribosomes are involved in using RNA for protein synthesis, but not directly holding DNA. Does that make sense?
So ribosome don't hold DNA. They are just the protein synthesis one, which is the rough endoplasmic reticulum.
That's absolutely correct. Ribosomes are primarily involved in protein synthesis, reading the messenger RNA to build proteins, and they don't hold DNA directly. Is there anything specific you'd like to know about?
Okay, and for DNA, nucleic obviously have DNA. Cytoplasm is of liquid state, so he holds the DNA. Mitochondria is the powerhouse, so he needs some DNA type thing to function. Then chromoplastic also have some amount of DNA, chloroplast. But ribosome only have your RNA. He don't have DNA. He function on like your what is that? Uh, Protein synthesis and all that. Rough endoplasmic reticulum. Ribosome only have uh, RNA. Then nucleus. Nucleus was Nucleus was like it is the brain of the cell, and this was discovered in your 1831 by Robert Brown. And like nucleus inside is this, in which there is a thread-like structure. This thread-like structure is called as chromatin. 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 This opens up in arranged as chromosomes. Opens up and arrange linear chromosomes, DNA plus RNA. So can you tell me what does this line actually mean? Like I know that nucleus inside is a thread-like structure which is called as chromatin, and now here it is written as open up and arrange linear chromosome DNA plus protein.
That line describes how chromatin changes during cell division. So you're right, chromatin is the thread like structure in the nucleus. Before a cell divides, this chromatin condenses and opens up to form the visible chromosomes. These chromosomes are linear structures made up of DNA tightly coiled around proteins. This arrangement is important for making sure that each new cell gets the correct copy of the DNA. Does that make sense?
So chromatin is thread-like structure. When this thread-like structure appear, the cell is about to do cell division, I guess, and it arranged linear, so it could divide properly the DNA and plus protein. That's the whole thing.
Yes, that's correct. The chromatin only becomes visible as condensed chromosomes when the cell is preparing to divide, ensuring the
So cell inside here is inside the nucleus. Nucleus inside is usually thread-like structure. Chromatin. This usually appears when the cell is about to do cell division, and when the cell division is about to happen, these chromatin usually have to be pretty linear to be able to provide protein and the right amount of DNA to the other cell. This is a PYQ. The nucleus is RNA plus ribosome protein factory. So from nucleus, this protein is generated from RNA and your ribosome. Now the deoxyribonucleic acid DNA genetic material genus is found in nucleus, mitochondria and plastid. Semi-autonomous called cell organelle. So no, the DNA deoxyribonucleic acid is a genetic material genus found in nucleus. This DNA is found in your nucleus, obviously where it is produced. Nucleus, mitochondria, because it is the part of the house of the cell and it needs some DNA to produce process correctly. And then your plastids. Is plastid the thing which is like fills the 90% of the cell or something? Can you tell me, is plastid the same thing which is the one which fills like plant cell 90% like the, or is it called vacuoles?
No, plastids and vacuoles are different things. Plastids like chloroplasts are involved in functions like photosynthesis and they can be different colors. The
Okay, that's
large
the,
vacuole,
okay, so vacuole is the one which fills, right?
yes,
투수 투수하고 응? 나도
So nucleus would have DNA because it produces this. Mitochondria is because he's the one which is managing everything in the cell. Plastid is like chloroplast, chromoplast, like they are have also function like functioning of like green pigment and all that. So it also have that DNA. Now RNA is ribonucleic acid. In rare RNA is genetic material. Example HIV. Okay. So there is a difference in RNA and DNA. Obviously, everybody know. I'm not gonna do much on that. But RNA one strand, but RNA have a special thing which is uracil, and this has thymine DNA. I always run these two only because everything else is same. They all have similar thing, but uracil is in RNA and thymine is in DNA. The nucleoid undefined nucleus region in the prokaryotes, not enclosed by a membrane like prokaryote. Then there is your nucleoid. Nucleoid is the undefined region of a cell. Defined nucleus or something like that, which is in prokaryote. Endoplasmic reticulum, Cather Potter, endoplasmic reticular. Information of tubule crystal found near of nucleus. Rough endoplasmic reticulum, rough protein synthesis. Smooth endoplasmic reticulum, flat, and lipid synthesis don't have ribosome. Vertebrates living smooth and toxic, so detoxifying. Okay, smooth endoplasmic reticulum because it has WBC, I guess. Yeah, WBC would be like different thing. Detoxifying is from different. WBC would be talk when it is like uh, fighting from your bacteria and all that. Okay. So this does this protein synthesis. This does lipid and fat synthesis. The mitochondria, semi-autonomous, inner membrane crystalline, inner membrane large surface area, outer membrane pores, fluid and matrix. Help in cellular respiration, ATP procedure, nutrient into energy. ATP, carrying of energy, ADP, androstenedione diphosphate, glucose plus O2 cell mitochondria. Christine. membrane do have pores to fluid and matrix as their large surface area and membrane, and this help in cellular respiration by ATP, uh, energy current, like bracket is add energy ATP प्रोड्यूस करते हैं, nutrient into energy. So powerhouse of the cell, basically it is giving the battery, you could say. Yeah, the plastid one which have DNA. Now we are going to talk about that. So these plastid obviously are like chromoplast and your like leucoplast. So these are not in animals. This is a basic knowledge, I would say. Uh, and this discovered was by Aaron Haeckel, Arrest Haeckel, Arrest Haeckel, in 1866. Arrest Haeckel. Found in plastid found. Then found in plant double membrane semi-autonomous DNA and ribosome protein. Yeah, these have protein and DNA. Plastid, mitochondria, and your nucleic acid. Then chromoplast, colored pigment. There is your carotenoids, which is caxtotin and xanthophyll. So caxtotin is orange and red, xanthophyll is yellow. Then there is a special pigment is chlorophyll, which is chloroplast, sorry, which is your green kitchen of the cell, obviously. Then there is leucoplast, which is colorless. Starch is produced by amyloplast, oil is alleloplast, and protein is proteinoplast. So I would say it is pretty easy to learn if you know what you're seeing at. Okay, so the first is your chromoplast. This is the colored pigment. Colored pigment. In this, it is like your colored pigment, and you could say that this have two thing. So first thing, sorry, it has like colored pigment. So this colored starts from your Keratinoids are of two types. First is your uh, caxtophil, cax, caxto, caxtonian. Second is your xantophil. So caxtophil is in your, like, you could imagine like a cactus, right? This is a cactus and this has spikes, right? And this might hurt you, so you would become red or you could become orange. Red or orange you could become. And the xantophil is your special one, which turns you into yellow. Like you can imagine like your caxtophil is you're getting attacked by cactus and you're getting red and orange. Xantophil is like xantophil, it is yellow. Fika, you could imagine like that. Then this is that caxtophil and xantophil, and this is a PYQ. And the special pigment which is clo chloroplast, you everybody know this, that this is green kitchen of the cell. So they would not ask this thing, they would ask caxtophil and xantophil in your chloroplast, in your plastids. So plast like CHLOROPLAST, plast and plastid, you could imagine as that. Now there is one more which is leucoplast, and this is more important. This is the colorless pigment. Colorless pigment. And this leucoplast help in storage too. In storage of, like, if we are talking about starch, starch is amyloplast. Then your oil is your Eleoplast, like oleoplast, you could say. Then protein is your proteinoplast. So the whole thing comes up to plastid. Plastid have DNA and your protein. Okay, it was discovered by Aaron Haeckel. Now, plastid, there is your chloroplast. Chloroplast, there are some colored pigment. In colored pigment, there is your caxtoplast, which is red and orange. Then there is your caxtoplast, and then there is your xantophil. Xantophil is yellow. Then there is your leucoplast. Leucoplast helps in storage. If storage of starch you are talking about, then it is amyloplast. And if you're talking about like storage of like your oil, then it is your eleoplast, or you could say oiloplast. Then if you're talking about your protein, then there is your proteinoplast. So this guy is in this or oranges, then this guy is in this. You could imagine starch amyloplast, and this is a PYQ amyloplast, and oil is your elioplast, this is E, then protein is your proteinoplast. Then there is your Golgi apparatus, Camilo Golgi, also known as Golgi bodies or apparatus. The icostate, endoplasmic reticulum, parallel tubes, pouch vessels, cis phase, trans phase. Trans is that, cis is above one. Okay. Synthesis of lysosome, making of lysosome, Golgi body. Function of Golgi body: transporting, patching, and synthesis. So double membrane is only for to protect the important stuff, right? So like your nucleus, like it has nucleic membrane and all that. Nucleus, mitochondria, because it is the battery of the cell or powerhouse of the cell. Then there is your plastids like chromoplasts, leucoplast, caxtoplast, xanthophyll. These are only the three one which have this. And the only stuff which doesn't have this membrane is ribosome, the protein one. It doesn't have any membrane because it is like attached with like endoplasmic reticulum and just move freely with them. So ribosome don't have membrane. Then there is your single membrane. Single membrane is on endoplasmic reticulum, not a rough. Rough is like attaching to ribosomes. That's ribosomes are attaching to the endoplasmic reticulum, which have the membrane. But like this is the endoplasmic reticulum. The rough one is just attaching to it. So while you're attaching, you don't need the membrane. So the endoplasmic reticulum need a membrane because it is carrying the rough, which is a ribosome. Reticulum. Then Golgi bodies. Golgi bodies are like transportation and all that, synthesis and all that, protein and all that, transporting, packing, synthesis, making ribosome. Golgi bodies. Nucleus protein histostones are highly alkaline protein that helps in packing DNA into structural unit of nucleus. हिस्टोस्टोन कहते हैं न्यूक्लियस में हिस्टो हिस्टोन्स बल्कि बॉडी lysosome vacuoles. Now, lysosomes are the membrane pouch-like structure filled with digestive hydrocyte enzyme. Worst first management of cell. Lysosome best when the cell is not functioning properly. Suicidal bag of the cell and synthesizes rough endoplasmic reticulum. Does ribosome synthesizes the rough endoplasmic reticulum?
It's the other way around. Ribosomes are attached to
No,
the
no,
rough
no.
endoplasmic.
No, wait. What I was want to say, the lysosomes are the one which synthesizes the rough endoplasmic reticulum.
That's also not correct. Lysosomes are mainly involved in breaking down waste materials and old organelles. The endoplasmic reticulum, both rough and smooth, is synthesized by processes that involve various components of the cell, including ribosomes but not lysosomes directly.
Brother, in endoplasmic reticulum, the rough is part is ribosome, which is protein. How is protein breaking down itself?
Ah, I see your confusion. The ribosomes on the rough endoplasmic reticulum don't break down proteins, they actually synthesize them. The proteins are made by the ribosomes and then processed within the ER. Breaking down proteins is mainly the job of lysosomes or proteosomes, which are different parts of the cell.
Then I have written here enzyme only synthesized by RA reticulum contain protein. Is this wrong?
Yes, that's not completely correct. While many enzymes are proteins and some are synthesized by the rough endoplasmic reticulum, not all enzymes are made that way, and not only the rough ER synthesizes them. Also, enzymes can break down substances, but the ribosomes themselves don't break down proteins.
Then there are vacuole, sac, pouch-like structure, single membrane, animal, small and transparent. Yeah, vacuoles are those one. Like this is the cell, and in plants, this is I'm talking about. These are the big ones. So these almost take 90% of the plant cells area, and these are like major role is up to 90% space below the cell. Provide rigidity and turgidity. Advanced vacuole contain tomoplast. Then there is your pre-Aximus small vessels. Deoxifies reactive oxygen species. Then there is your centosis. In cell division, helps in movement of plant. Small organisms help in clipper from centrocells. Centrioles helps in movement of these cells in the cell divisions. Centrioles. Plasmodesmata. This centralis movement is only in the animal. because those are the living one. If they had this movement kind of thing in the plant, then plant would be moving, I guess. Can you tell me what is this tonoplast, which is present in like plant cell and absent in your animal cell?
The tonoplast is the membrane that surrounds the large central vacuole in plant cells. It plays a critical role in maintaining turgor pressure, which gives plants their rigid structure and shape. It also actively transports various substances between the vacuole and the cytoplasm, helping to regulate ion balance and store nutrients. Animal cells don't have a central vacuole, which is why the tonoplast is unique to plants. Does that make sense?
So the tonoplast are only present in plant because they are the like this is the plant cells. So obviously this is the nucleus at the uh, not at the center but at the corners, and there is some rigidity on the corners too. That is also something I remember that just now. uh, so like this is vacuoles, right? So vacuole outside is tonoplast. This helps the chloroplast, which is outside jelly like substance in the cell, to this tonoplast helps in transportation in the inside of vacuole and outside of the vacuole, and this provide turgidity and the shape of the cell basically. Centrolis is absent because centrolis is the thing which we do when we have to do the cell division, and cell division is not that common. It is common in like the growing one, uh, growing cells, but not in the movement of this type of thing. That is not really present there. uh, yeah. Meiosis, mitosis, phosphate, promphase, metaphase, anaphase, telophase, ppmat, ppmat. Then the cell division, which is the cent. What the heck? Centrotriolus is. This is the guy which the movement of the purposes of cell division is growth, repair, gamete formation. So first is the mitosis, Tom Fleming and Tom John Farmer Morrow. So can you tell me what is the meaning of this? Like I'm studying the cell division, it is saying mitosis, Tom Fleming, the meiosis, Tom John Farmer and Moore.
It means that Walter Fleming is credited with coining the term mitosis, and John Farmer and J.E.S. Moore coined the term meiosis. These are the scientists who introduced these terms to describe the different types of cell division. Does that make sense?
So mitosis is like one cell is there and it is dividing into two cell, normal type of cell division. Then meiosis was discovered by John Farmer and Moore. Farmer and Moore, Moore, Moore. John Farmer and Moore. He discovered these. Like in this one cell goes into one cell goes into like 4 division. And like in the mitosis, which was discovered by your what is his name, Fleming, Fleming, John Fleming, not John Fleming, it is Fleming. He discovered mitosis. He tells what is mitosis as. Now there are processes here. So first is this, which is prophase, just preparing. Second is like, you know how I told you in the nucleus, the chromatin or something like that. The chromatin is become thread-like structure when it about to do the cell division thing, and it become linear. So that type of phase happens here, which is prompt phase. So they are doing prompt here. Then this is the metaphase, which they are about to do it. They are just prominent now, they are at mature now, metaphase. Then there is anaphase, which is just gonna do it. And then there is your telophase, which is about to do it, just happened about. The nucleus division is cytokinesis to cytoplasm. Nucleus divides, cytokinesis to cytoplasm. The nucleus divides to cytoplasm in cytokinesis. and you can remember this as PPMAT. First is prophase, then promphase, then metallic phase, then anaphase, then telophase. PPMT for mitosis, mitosis, which was discovered by Fleming, and meiosis, the farmer, and your Moore. And the meiosis is like there is 2n here nucleus. It discovered into nucleus. Now this nucleus get discovered in the divided into 2. So like one cell is doing 4 here. That's how they did it. Does this thing. The 4 what I was talking about, cell division. So nucleus, then cell wall, endoplasmic reticulum, then vacuoles.
सब ज्यादा कंटेंट अच्छा तुम मेरी तेरे को बुला सकती है